Hello, my name's Kate McElroy and I'm one of the Otago Medical Research Foundation summer studentship students, and I've been funded by the Ailsa Goulding Award. My project is going to be looking at CRTC1 mammal2 mutations in salivary gland mucoepidermoid carcinoma in a New Zealand population. Mucoepidermoid carcinoma is the most common salivary gland malignancy in both adults and children. All salivary gland malignancies look very similar under a microscope, which makes the pathologist's job very difficult. The clinical implication of this is that when these cancers aren't accurately diagnosed, the prognoses are very different for the different types of cancer, and the treatment approaches are very different.
A recent discovery that's very exciting is the identification of a specific gene associated with this tumour. Now, this gene is the mammal2 gene, and when it mutates, we're able to stain this in the laboratory using fluorescent dye to identify that the gene is no longer intact. When you observe the diagram provided, you'll see that when the cell is normal, you'll see only a single greeny-yellow light occurring at the gene locus. However, when the gene is mutated, you will see that the signal becomes split into a green signal and a yellow signal.
However, none of this research has been done in the New Zealand population, and so at this stage we don't know whether mucoepidermoid carcinomas in our population carry this mutation. If we do carry it, how many of these cancers are positive for it? Whether or not our original diagnosis was accurate. These are some really important questions that we're going to be looking at investigating, looking at 20 years worth of mucoepidermoid carcinomas that have been diagnosed at the Dunedin Oral Pathology Centre.